The laboratory report came back in two parts. The first part matched what the researchers expected, the second did not. In the spring of 2017, a research team at Northwestern University published results from a genetic study conducted in an Old Order Amish community in Berne, Indiana. The subjects were members of a church district whose families had farmed the same flat agricultural terrain since the 1850s, speaking Pennsylvania Dutch at meals, working the fields with draft horses, and drawing their marriage partners from a closed circle of surnames any county recorder could have traced back 4 or 5 generations without lifting a pen. The researchers came looking for a specific inherited variant, a mutation in a gene called SERPINE1, which governs production of a protein the body uses to regulate blood clotting and the rate of cellular aging. What they found in the genomes of these farming families was a mutation that appeared at meaningful frequency in almost no other recorded human population in the world. The carriers were living longer, they moved through their 80s with a cardiovascular profile that looked decades younger than their recorded age. Their arteries showed less stiffening than age-matched control subjects. Their blood metabolized insulin at rates the mainstream American population had not sustained in 3 generations. Their telomeres, the protective end caps on chromosomes that geneticists use to estimate biological age, were measurably longer than those of any non-Amish comparison group the research team could locate. If you're enjoying this story so far, don't forget to hit the like button and subscribe. Drop a comment telling me where in the world you're watching from. I love hearing from you. The study appeared in a publication called Science Advances. Science journalists covered it briefly, and then coverage moved on. What the Berne study had found was not a curiosity attached to a marginal population at the edges of modern life. It was a window into what happens when a founding genetic population closes its borders, holds them closed for 300 years, and preserves inside its own genome a set of inherited variants the broader American population had gradually lost as it dispersed across a continent. If you have ever traced a family line to a German-speaking immigrant who arrived in Pennsylvania between 1710 and 1850, a probability exists, small but real, that your family's genealogical record and the Amish community's genetic record share a common ancestor. The connection predates the denomination. It is older than the name Amish itself. And what the DNA extracted from those Indiana farms tells us about the growing genetic distance between the founding European population and the modern American genome is a story the census records were never built to carry. To understand what was found in those Indiana genomes, you have to understand who the Amish are, and more precisely who they were before they became Amish. In the summer of 1693, a Swiss Anabaptist bishop named Jakob Ammann convened a series of meetings in the Alsace, the disputed border territory between France and Germany, along the Upper Rhine, where Anabaptist communities had maintained a presence since the middle of the previous century. Ammann belonged to a tradition that had been under sustained institutional pressure across Central Europe for close to 150 years. The Anabaptist movement had broken from the mainstream Protestant Reformation in the 1520s over a cluster of theological positions that made its members politically inconvenient to every civil authority they lived under. They refused to baptize infants, holding that baptism was a covenant entered by conscious adult decision and not a sacrament administered at birth without the recipient's knowledge. They refused to swear oaths of loyalty to civil governments. They refused military service. In an era when the authority of every European state rested on a combination of religious conformity, military obligation, and the legally binding oath, the Anabaptists rejected all three. The institutional response across the German cantons, the Rhine-Palatinate, and the Swiss Confederation was sustained and systematic. Governments expelled them from territories, confiscated their land and property, imprisoned their community leaders, and in the most intense periods of the preceding century had executed a substantial number of them for the offense of their convictions. The communities survived by relocating repeatedly to the high valleys of the Swiss Bernese Oberland, to the margins of the Rhine-Palatinate, to the Alsatian lowlands, wherever a local count or a Protestant ecclesiastical authority was willing to accept their agricultural labor on difficult terrain in exchange for leaving them in peace. And they moved again whenever that peace expired. The communities that survived through these successive displacements were small, geographically isolated from one another for long stretches, and accustomed to forming marriages within a limited circle of families who shared both their theology and their legal exposure. The social structure that made survival possible in that environment was simultaneously a genetic one. The gene pool that traveled with those congregations through 2 generations of European displacement was narrower than the broader Swiss or Palatine population from which it had originally drawn its founders. The Anabaptist communities in the Alsatian cantons had been forming marriages among themselves long enough by 1693 that their collective genetic profile reflected not the full diversity of Central Europe, but only the particular fraction that had passed through the filter of a persecuted minority community sustained by internal marriage across 3 or 4 generations. What Ammann brought to his 1693 meetings was a demand for stricter separation from the surrounding world than the broader movement had been observing. He called for more consistent enforcement of the Meidung, the practice of shunning members who had violated communal standards or broken from the Church's discipline. When the existing Anabaptist leadership in the Swiss cantons refused, Ammann's faction separated from the main body. They took the name Amish from their bishop, and they carried that name and that commitment to separation into the following century, when the accumulated pressure of European religious politics finally sent the first communities west toward the Atlantic. The crossing itself reinforced what European persecution had already produced. A congregation that sailed together from a port in Rotterdam or Amsterdam in the early 18th century arrived in Philadelphia already constituted as a community. They had shared the voyage, the provisions, the confinement, and the deaths that typically accompanied a 7 to 9 week Atlantic transit in a vessel that offered no real medical care and minimal separation between families. The communities that came ashore in Pennsylvania were, in every social and organizational sense, already the communities they intended to be in America. The genealogical network they brought was not something assembled after arriving. It was carried intact across the Atlantic and planted as a complete structure in the farming valleys west of the city. The individuals who might otherwise have entered the community from the broader Pennsylvania German population around them were not invited in. The community looked inward from the moment it arrived, and the gene pool it carried looked inward with it. The first significant wave of Amish immigration to North America arrived in Pennsylvania beginning in the 1720s and 1730s. They traveled as part of a much larger Pennsylvania German migration that moved somewhere between 150,000 and 250,000 German-speaking people through the port of Philadelphia across the course of the 18th century. Palatine Germans, Swiss Reformed settlers, Mennonites, Lutherans, and Dunkards all moved through the same geographic corridor, filling the limestone farming valleys west and south of Philadelphia with communities that maintained their language, their religious structures, and their social organization across several generations of American settlement. Within that larger movement, the Amish did not arrive as scattered individuals seeking agricultural land. They arrived as communities. They settled adjacent parcels, maintained the congregational structures they had organized in the Alsatian valleys they had left, and continued the marriage patterns those valleys had established over the preceding generations. The surnames visible in Lancaster County and Somerset County records from the mid-18th century reflect the geographic origins of these settlers with a precision that has made those records useful to researchers ever since. Byler, Stoltzfus, Yoder, Zuck, Hostetler, Lapp, Blank, Byler, King, Miller. These are not a random sampling from the German-speaking world. They are the documented surnames of specific families from specific valleys in the Bernese Oberland, the Rhineland-Palatinate, and the Alsatian lowlands, families that had already been forming marriages within a tight genealogical circle for 2 or 3 generations before the Atlantic crossing. The crossing did not broaden that circle, it compressed it further. The community that arrived in Pennsylvania was a founder population in the most precise genetic sense, a small subgroup drawn from a larger European source population, carrying a genetic profile shaped not by the full diversity of that source, but by the fraction that had survived the filter of European persecution and made the crossing together, bound by the same faith and the same social structure they had maintained under pressure for a century before they sailed. By the first United States Census in 1790, Amish communities held established ground in Lancaster and Somerset counties, in portions of Maryland and Virginia, and in the earliest Ohio settlements. Their numbers were modest against the broader Pennsylvania German population that surrounded them, but their internal genealogical structure was already a defining feature of how the community organized itself. Marriage occurred within the community, between Amish individuals, from a pool of surnames that a competent genealogist can today trace across 3 centuries of American settlement without losing the thread. The surnames in 1790 are largely the same surnames visible in Amish genealogical records 2 centuries later, connected by an unbroken chain of marriages that researchers can follow in many family lines without encountering a gap. Amish communities have maintained their own genealogical records separately from state vital registration systems for most of their American history, and several Old Order settlements have compiled these records into comprehensive published family books that document births, marriages, and deaths across the full span of a community's American presence. For population geneticists, those volumes function as the paper counterpart to the DNA archive the community carries in its own genome, a cross-referenced, multi-generational record that allows researchers to connect a specific genetic variant in a living individual to a specific founding family from a specific decade in the colonial period, with a precision that is almost never available in open population research. The closure deepened in the 1870s, when a dispute over the adoption of new technologies divided the American Amish Church into progressive and conservative branches. The conservative branch became the Old Order. They declined the telegraph, certain farm machinery, and rail connections, but the deeper refusal was the broader social opening those technologies represented. The Old Order organized community life around a system of church districts, each comprising a group of families who met for worship in each other's homes, and whose members drew their marriage partners from the same closed genealogical pool the community had been maintaining since the colonial period. They did not admit converts from outside. They did not permit marriage with non-Amish individuals. The gene pool that had been narrowing since the Alsatian refugee communities of the 1690s continued to narrow with each generation, and the mathematical consequences of that narrowing accumulated in the biological record the community carried. In a population that admits no new members and prohibits outmarriage, a rare genetic variant can rise from low frequency to markedly higher frequency over a small number of generations without any external selective pressure. A variant carried by 1 in 100 people in the founding European population can reach 1 in 20 within the closed community across 4 or 5 generations, because every carrier is descended from the same small founding group, and the variant circulates through the marriage network rather than dispersing outward. The founder effect is the term population geneticists use for this phenomenon, and it operates with particular force in populations that are small, closed, and genealogically documented in detail. By the 20th century, the Old Order Amish founder effect had produced one of the most extensively documented cases of genetic variant concentration in any non-island human population. Ellis Van Creveld syndrome, which affects skeletal development and cardiac structure and results from mutations on chromosome 4, appears in Lancaster County's Old Order Amish at a frequency roughly 200 times higher than in the general American population. The Lancaster cases trace to a single founding couple who arrived in Pennsylvania before 1770, whose descendants have been marrying other descendants in the same county for close to 3 centuries. Glutaric aciduria type 1, which disrupts amino acid metabolism and requires careful clinical management from early childhood, reaches frequencies in several Ohio Old Order communities that are almost undetectable outside them. Maple syrup urine disease, which prevents the normal metabolism of branched-chain amino acids and requires dietary management from earliest infancy, concentrates in specific Pennsylvania church districts at rates that have made those communities the primary clinical population for studying the condition's long-term natural history and its response to current treatments. These concentrations are the visible cost of the founder effect, disease-associated variants that the founding families happen to carry, held at community-level frequency by 3 centuries of closed inheritance. They are also only one face of what the Old Order genome preserved. The other face was what Douglas Vaughn's team found when they drove out to Indiana with their sample kits and their laboratory assay protocols and a list of church district families who had agreed to participate in the study. If you're hooked on this story, make sure to subscribe and turn on notifications so you never miss an upload. Your support means everything and helps me bring you more emotional stories like this one. The SERPINE1 gene produces a protein designated PA1, plasminogen activator inhibitor 1, which participates in the regulation of blood clotting and in the broader biochemical process of fibrinolysis, the mechanism by which the body dissolves clots after they have served their purpose. SU1 operates within a regulatory network connecting the coagulation system to metabolic function and to the maintenance of the cells lining the blood vessels. In clinical populations, elevated PAI-1 is associated with increased cardiovascular risk, with insulin resistance, and with the progressive arterial stiffening that researchers use as an index of biological aging in the vascular system. Dysregulation of this protein in either direction carries consequences that clinical researchers have been documenting across multiple disease categories for several decades, and the system it regulates sits at a convergence point between cardiovascular health, metabolic function, and the cellular aging process. The variant the Berne community carries is a 4 base pair deletion in SERPINE1 that reduces PAI-1 production below typical levels. Before the Vaughan study, this deletion had appeared in the published literature only in isolated case reports, identified as an incidental finding in patients being assessed for unrelated clinical conditions. No research group had previously found it concentrated at population frequency in any human community. Vaughan's team tested individuals from the Berne Old Order Church District and found the deletion in approximately 1 in 4 of those tested. In the general American population, the best available estimates place the deletion's frequency below 1 carrier in several hundred. In Berne, it appeared in 25% of the community. The published findings were specific. Heterozygous carriers, people who had inherited 1 copy of the deletion alongside 1 normal SERPINE1 copy, showed a cluster of metabolic markers distinguishing them from non-carriers in the same community. Their fasting insulin levels were lower than those of age-matched non-carriers in the same church district. Their PAI-1 activity, measured directly from blood samples, was reduced in proportion to their carrier status. Their telomere length was greater than that of non-carriers of the same age living in the same community under the same conditions. Their arterial stiffness, assessed by pulse wave velocity, was lower than in both the non-carrier Amish group and the non-Amish control population drawn from the surrounding county. The Berne community's average lifespan exceeded the county average for the surrounding non-Amish population by approximately 10 years. Researchers treat this figure carefully. The lifestyle characteristics common to Old Order communities, sustained physical labor and agricultural work, low rates of tobacco and alcohol use, the dietary patterns that follow from Old Order farming life rather than from industrial food supply chains, and the cohesive social structures maintained through the church district system all contribute to that gap in ways the available data cannot fully separate from the genetic contribution. But the specific metabolic markers tied to the SERPINE1 deletion persist when lifestyle is held constant within the community. Carriers in the Berne church district, living and working alongside non-carriers under identical agricultural and dietary conditions, still show measurably different metabolic profiles from their neighbors without the deletion. The variant carries an effect that the lifestyle context alone does not account for. The age structure of the Berne community, visible in the church district's own genealogical registers, is consistent with the metabolic findings. Berne families show a higher proportion of individuals living past 80 than the surrounding non-Amish population, and a lower incidence of the cardiovascular and metabolic events that most commonly curtail productive life in the general American population in the 7th and 8th decades. Researchers who visited the community during the study period noted that the district's oldest members were not living in the kind of managed decline common to age-matched non-Amish Americans, but in active agricultural engagement, still working land and participating in the community's daily labor at ages most American medical researchers treat as benchmarks for exceptional longevity. The question the study left open was why the Berne deletion had reached its current frequency. Standard population genetic theory offers drift as a partial answer, the random fluctuation of allele frequencies in small populations that can carry a rare variant upward through generations without any selective pressure. Drift is real and operates in every small closed community, but drift alone does not reliably predict 1 in 4 in a population with the Berne church district's documented size and genealogical depth. The more consistent explanation involves a selection component, the amplification of the deletion's frequency through the accumulated reproductive advantage of people who live a decade longer and maintain better metabolic function across their adult years. Carriers reproduce over a longer span of years. Their children inherit the deletion. The community's closed marriage structure keeps those children inside the same genealogical pool, and the frequency rises with each generation. The genealogical registers the Berne community maintains, which document each birth and marriage in the church district with the precision the Old Order requires in lieu of interaction with state vital registration systems, allow researchers to trace the deletion through the families carrying it to a cluster of original immigrant surnames appearing in Pennsylvania German church records from the 1730s and 1740s. Those surnames trace in turn to the Alsatian lowlands and the Swiss Bernese Oberland, the same regional cluster from which the early Amish movement drew its founding families after Ammann's 1693 schism. Somewhere in that genealogical chain, a man or woman born in a Swiss valley or on the Alsatian plain carried the four-base pair deletion. That person sailed to Philadelphia in the 1720s or 1730s. Their children married other children from the same founding circle in the Lancaster County settlement. Those grandchildren did the same, and the next generation after them, and the 10 following that. What had been a rare variant in one corner of 18th century Europe became, across 300 years of documented closed inheritance, a feature present in 1 in 4 people in an Indiana farming community that has no living connection to the world that originally produced it. The mainstream American population, descended from the same broad Northwestern European immigrant stock, but mixing outward through 3 centuries of open marriage, westward migration, and demographic expansion across a continent, carries no detectable trace of this deletion at any measurable population level. The dispersal was complete. Whatever the founding colonial-era European immigrants brought to Pennsylvania in the 1720s and 1730s, including the variants associated with slower vascular aging and better late-life metabolic function, spread through a gene pool too large and too mobile to hold them at any frequency the available testing can currently detect. What survives in Berne survives because the community around it stayed still. The Clinic for Special Children in Strasburg, Pennsylvania, which D. Holmes Morton founded in 1989, was built to address the other consequence of the same genetic isolation. Morton developed a community-based genetic testing network for the Lancaster County Old Order population, integrating the community's genealogical records with molecular genetic testing in ways that gave families accurate information about carrier status for the rare recessive disorders the founder effect had elevated to high frequency. The clinic's methods, built on the intersection of community trust and genetic science, became a model for founder population medicine that researchers in the Hutterite communities of the Northern Plains, the Old Order Mennonite settlements of Ontario, and the island populations of Iceland and Sardinia have since adapted for their own purposes. What Morton built over 3 decades in Lancaster County, and what the Vaughan team confirmed again in Berne in 2017, points toward a conclusion about genetic closure and open mixing that the mainstream American conversation about ancestry rarely stops long enough to examine. The Old Order Amish in the 21st century are the closest living approximation to the Swiss and Alsatian founding population of the early 18th century that exists anywhere. The mainstream American population is not that. 300 years of the largest voluntary migration in recorded history, incorporating Irish famine survivors, Italian and Southern European contadini, Ashkenazi Jewish refugees from Eastern Europe, descendants of the West African transatlantic slave trade, Mexican border communities, and waves of Korean, Vietnamese, and East African arrivals through the mid-20th century displacement crises, have produced in the general American population a genetic diversity the founding colonial-era settlers could not have anticipated. The Amish genome and the American genome are now, by any research standard, samples drawn from 2 separate population histories. They share a founding era and a founding geography in the Pennsylvania German counties. The genetic record beneath those shared surfaces describes a separation so thorough that the 2 populations are treated as distinct genetic entities in research literature, which is exactly what they have become. The divergence between the 2 genomes has practical consequences beyond what the Berne study measured. Pharmaceutical researchers developing treatments for cardiovascular disease and metabolic syndrome typically test those treatments in populations that reflect the general American genetic profile, which is the product of 3 centuries of open mixing among dozens of source populations. That genetic heterogeneity makes it difficult to isolate the effect of any single variant on a complex disease process. The Old Order Amish, because they represent a comparatively homogeneous founder population with documented pedigrees and stable environmental conditions, have become useful models for studying the specific effects of individual variants on disease processes that are difficult to isolate in the open population. Several research groups now maintain ongoing relationships with Old Order communities in Pennsylvania and Indiana for exactly this purpose, not because the Amish genome represents the American genome, but because it is the furthest possible thing from representative, and that contrast is what makes it scientifically useful. This matters for people who carry no Amish surname and no Amish ancestor in their documented genealogy. The 4-base pair deletion that 1 in 4 Berne residents carries is not an exclusively Amish variant. It is a human variant that survived inside an Amish community because that community stayed closed. The deletion is almost certainly present in some of the families descending from the same 18th century Palatine and Swiss immigrant wave, dispersed through 3 centuries of open marriage into the broader American population, but at a frequency so low that it produces no detectable population-level signal and does not appear in any consumer testing panel currently offered. Most commercial DNA tests do not report on SERPINE1 variants. They are not included in the standard panels any major ancestry testing company offers its customers. Clinical genetic testing that would identify the deletion is ordered in the context of coagulation disorder evaluations, not in the context of longevity research or ancestry analysis. For the consumer who sends a saliva sample to one of the large commercial testing platforms, the Berne deletion is not in the results. It sits below the reporting threshold of every panel on the market. That invisibility is structural. Commercial DNA testing is calibrated around the variants present at meaningful frequency in large populations, because those are the variants that generate results useful to enough customers to justify their inclusion. Variants present in less than 1% of the general population do not meet that threshold. The founder effect signals, the variants that survived at high frequency only in specific closed communities and dispersed to near zero in the open population, belong to the category of genetic information that current consumer testing was not built to retrieve. What a consumer receives is an accurate account of what the test was designed to find. It is not an account of the full genetic record the sample contains. The deeper record, the one that connects to the founding European populations before 3 centuries of open mixing carried it below current detection, is present in some of those samples and inaccessible in all of them. What the Old Order Amish genome has become is a living archive of a specific moment in European genetic history, held intact because a community decided, for theological and cultural reasons entirely unrelated to genetics, to remain separate from the surrounding world across 300 years. The researchers who examine this archive are not making arguments about population hierarchies or comparative merit. They are asking what happens when a founding group stops mixing and what stillness carries across 10 or 12 generations. The Berne study gave one answer: a deletion associated with slower cellular aging, better late-life metabolic function, and a measurable extension of average lifespan, preserved at 1 in 4 frequency in a community of farmers who do not use the word telomere and would not have considered it relevant to how they live. What survived in those Indiana telomeres and insulin receptors and 4 base pair deletions is a record no paper archive and no genealogical database was designed to hold. It survived not because anyone set out to preserve it. It survived because the community around it chose, for 300 years, to stay home. And in staying home, they kept something intact that the broader continent's descendants had already scattered across too wide a geography for any current test to recover. That is what the Berne study found when it drove out to Indiana, with its sample kits and its assay protocols, and its expectations of a routine founder effect documentation project. Not a curiosity, not an anomaly, a genetic record from a founding moment in European and American history, still legible in the blood of people who do not use the vocabulary the science uses to describe it and never knew the record was there. Thank you so much for watching until the end. If you love this story, check out the other videos on your screen now. I think you'll really enjoy them. And don't forget to subscribe if you haven't already. See you in the next story.